Be focusing on Extremophiles by Tor Moncrief, Zach Friedlander, James Kerbo, and Trin Nugent. Extremophiles. Extremophiles are often microorganisms that are capable of thriving in extreme environments that would normally be deadly to other forms of life. The environments that they are capable of surviving include those of great diversity with extreme temperatures, pH, salinity, pressure, and other conditions. There are several different types of extremophiles, such as thermophiles, psychrophiles, halophiles, acidophiles, alkalophiles, and radiation organisms. Moving on to our first group of thermophiles. These organisms thrive in high temperature places where most other organisms' enzymes would denature. The temperature can range from up to 45 degrees Celsius to 122 degrees Celsius. They are found in environments like volcanic hot springs, geothermal vents, and deep sea hydrothermal vents. Thermophiles represent a diverse group of microorganisms including bacteria, archaea, and some eukaryotes. Thermophiles have several biotechnical applications due to their high heat resistance. They are studied for potential in astrobiological studies and the search for life on other planets with harsh conditions. Next up, we have psychrophiles. Psychrophiles are the opposite of thermophiles and are organisms that live in extreme cold. Psychrophiles include bacteria, archaea, fungi, algae, and some protozoa. These organisms have biotechnical applications due to their ability to function in these cold temperatures. Enzymes adapted for the cold can be used in food processing, pharmaceutical production, and cold active detergents. Moving along, we have haliophiles. These organisms are adapted to high salinity environments. They survive in environments such as salt flats, saline lakes, and hypersaline seas. Those that are hypersaline often live in low temperatures. These organisms are researched to help with water treatment. There are three types of haliophiles. One being moderate haliophiles. They grow in environments with salt concentrations of 3 to 15%. The second one, extreme haliophiles. They like concentrations of 15 to 30%. And the third being haliotolerant organisms. They do not require a lot of salt for their growth. Up next are acidophiles. These organisms thrive in high acidity environments that can form due to industrial factory and mining runoff or by volcanic hot springs. They are capable of maintaining their internal pH by keeping it close to neutral even in extreme environments. This group includes bacteria, archaea, fungi, algae, and protozoa. They produce acid-stable enzymes. For example, acidophiles produce enzymes with acidic isoelectric points to increase stability. Last but not least, we move on to our radiation organisms. These organisms withstand high levels of radiation. These organisms are usually found in places with elevated levels of radiation, such as waste sites. These organisms have incredible and efficient DNA repair systems capable of fixing damage caused by the radiation. Because radiation organisms are so good at DNA repair, they have a lot of applications in the world that can be studied. Their enzymes are good for developing crops and biofuel that are resistant to damage. Moving on to various adaptations. As the environment changes, so does our little microscopic friends. Extremophiles have evolved different adaptations that allow them to survive. These include enzyme stability, membrane adaptations, osmotic regulation, and metabolic adaptations. Moving to our first adaptation, we have enzyme stability. Enzymes are biological catalysts that accelerate chemical reactions. They are sensitive to their environments and changes in conditions. Extremophiles can produce enzymes that remain stable and functional in their extreme conditions. Moving along with enzymes, we see enzymes have an ideal temperature at which they function the best. The stability of extremophile enzymes at extreme temperatures is due to their unique molecular structure, which are adapted to withstand denaturation. Enzymes also have an ideal pH. Enzymes from different organisms may have different optimal pH levels and stabilities. Chemicals are also capable of denaturation. Modifications such as glycosylation or phosphorylation can enhance the stability of enzymes for extremophiles. Additionally, the presence of ions and cofactors can protect enzymes. Looking at our next adaptation, we see membrane adaptations. These organisms develop membranes within unique compositions that provide stability. The membranes protect them from the extreme conditions by blocking them out. Furthermore, we see membrane fluidity is crucial for cellular processes, including enzymatic activity. Organisms can adjust the fluidity of their membrane by altering the composition of membrane lipids. In cold environments, organisms may increase the number of unsaturated fatty acids in their membranes. Proteins located in the membrane play a role in cell signaling, transporting nutrients, and cell communication. Bacteria living in rich nutrient environments can express specific membrane transporters to take advantage and regulate the nutrients. Taking a look at osmotic regulation, this is the regulation most used by haliophiles. Osmotic regulation refers to the control of water and solute balance across membranes. These types of extremophiles thrive in high salt and low water conditions, having evolved with specific mechanisms for regulation. Haliophiles have two major strategies to cope with high salt concentrations. They accumulate compatible solutes such as amino acids like proline, sugars like sucrose, or polyols like glycerols, which maintain balance without affecting cellular processes. These solutes prevent water loss from the cells. They also modify cell membrane and walls by incorporating lipid species, such as archaeomembrane lipids, which reduce permeability to metal ions and water. Moving on, we see metabolic adaptations. Extremophiles often have specialized metabolic pathways. This allows them to thrive in their environments. The type of adaptation varies depending on the characteristics for the environment that they inhibit. Thermophiles have adapted to having heat-stable enzymes like the thermostable proteases for protein degradation. They may also have pathways such as oxidative tricarboxylic acid cycle or non-oxidative pentose phosphate pathways, which are adapted to function at higher temperatures. Acidophiles possess proton pumps that maintain pH homeostasis and keep the cytoplasm close to neutral. Haliophiles produce salt-inhibiting enzymes with increased salt tolerance to allow them to maintain activity in their environment. Lastly, we have structural adaptation. Proteins produced by extremophiles may have unique structural adaptations to help them survive and not denature in harsh climates. Extremophiles may possess appendages that enhance the ability to survive. Examples of this include proteinaceous surface layers in bacteria to provide heat protection. Many extremophiles can form biofilms which give them protections against stressors. And this wraps up our presentation. I hope y'all learned something. Thank you.